Hi, my name is Biol. It's 25 August 2025. This is my Unit 3 presentation. I'll be talking about Giorgio Bernardi's essay review, about how the non-coding DNA mystery and how he's kind of found an explanation for that. So what is the non-coding DNA mystery? So it's a known fact that the majority of our genome is made up of non-coding DNA. So it doesn't code for any proteins, amino acids that can have a function in our biochemistry. So a lot of people raise the question: is this so-called junk DNA kind of like a vestigial organ that's been left through evolution and just serves no purpose anymore? Is it selfish DNA? So one of the questions or estimations that people have had is that it's like a transposon of a repeating sequence, almost parasitic, and that it just repeats and doesn't benefit the host. And then finally in 2012, the ENCODE project got a little bit closer, and they said or gave the proposal that about 80% of the genome was functional in a biochemical capacity, that they have biological functions as functional elements, but that proposal wasn't very well received at the time. There wasn't really a hard accepted definition of functional elements, so it wasn't really well received. I thought they were using that term loosely, so it didn't really go anywhere. He took a bunch of research and work on isochores. Those are just regions of DNA that are composed of guanine cytosine, either GC rich or GC poor. Whether they're rich or poor will affect density, sequencing, positioning. But what he found was that these different regions are they have a role in chromatin folding and the actual structure, the architecture of chromatin, which is really interesting. So he took all that research and applied it with like modern 3D mapping mechanisms, and found that it had to do with folding motifs for chromatin, which is super interesting. It kind of goes against what we commonly accept, that protein is the center of everything. Protein has all these functions. DNA codes for protein, very protein-centric approach. This is kind of against that, showing that DNA almost supports itself. DNA is coding for its own architecture, its own compartmentalization, which is super interesting. And that's kind of his explanation, his approach that he's found to the non-coding mystery.